What I'd like to do is give a little bit of a history of what Joachim's just told you about the elucidation of this system biochemically, and then try to exemplify some of the ways in which a simple biochemical pathway might be modulated and transduced for the task of physiological oxygen homeostasis. And then I'll hopefully finish with what hopefully be a little bit controversial about drug discovery and the vagaries of Darwinian biology. So first, the history. Coming from Oxford, I'd like to start with Haldane and the famous expedition to Pikes Peak. People here will know that Haldane deduced that carbon dioxide levels were the main controller of respiration, but he also became aware that people would breathe more rapidly at altitude and considered that this was most likely to be related to the retention of acid in anaerobic situations. And this was the attempt to prove the hypothesis, since he believed, therefore, that the acid would be secreted by the kidney and corrected, and therefore that the increase in respiration would correct over a few days. That was his hypothesis, that it would the carbon dioxide level would return to normal as breathing stabilized. And what you see is clearly that was not the case. So his hypothesis was not supported, but for some reason he failed to make the link to the sensing of oxygen, which really should have been done, not least because of the work of the heroine of that expedition, Mabel Fitzgerald. Since she was a woman, she couldn't be a member of Oxford University, and since she was a woman, the boys became a little bit concerned about her being unchaperoned with them at the summit of Pikes Peak in a rather comfortable hotel. So what they did was send her away on her own to make measurements at the towns at intermediate altitude. This, of course, was the Wild West, the mining communities. She survived all that and made these accurate measurements, which I think are the first exemplification of the extraordinary sensitivity of hemoglobin and what we now know to be EPO to changes in oxygen. And it was that sensitivity that led to the idea that there would be a specific oxygen sensing mechanism. So the work of Alan Esloff, of course, established EPO as a hormonal regulator, and this was the task that interested us, how to go from EPO to oxygen. There was one particularly interesting experiment performed with total clarity but dubious ethics. The infusion of cobalt into normal volunteers with serious psychiatric diseases. It was done in a famous East Coast US hospital. They didn't kill anyone, but they did generate an impressive erythrocytosis, and that established that cobalt, which is not a metabolic poison, could do this, whereas cyanide, which is, does not have that effect. And that established the concept of oxygen sensing in the regulation of EPO, which was believed, because of the extraordinary sensitivity and interesting properties, to be a specific property of EPO-producing cells in the kidney. And that's what we set out to find. This was an early experiment in my lab, which didn't work. What we wanted to do was make an EPOMer by expressing T antigen behind the EPO control sequences. But along with work from the Zurich group, it did establish the nature of the EPO-producing cells as the interstitial fibroblasts. Now, as you've heard, I'm a nephrologist, and the kidney has all sorts of really interesting cells, the glomerulus, the tubules, and practically every one of these have been proposed to make EPO, except the rather unspecialist fibroblast that actually does so. And in retrospect, that should have been a clue to what was to follow. But our intention here was to make an EPOMA and grow a cancer and grow the EPO-producing cells and use those as a substrate for molecular studies. That didn't work, but fortunately, Franklin Bunn in Boston had derived some hepatoma cells, HEP3B, HEPG2, that do make EPO in culture in response to low oxygen, and we and Semenza and Jaime Caro were able to use these cells to define this little sequence at the 3' end of the gene that could confer regulation on a reporter gene, and that was the first step. Our intention was to move step by step from EPO outwards to the oxygen sensor. Now, we immediately realized this experiment had another potential. What would happen if those sequences were inserted into other cells that didn't make EPO? And after a few false passes at this, which arise because when you make these cells hypoxic, you don't absolutely know what you're doing. So sometimes your cultures are hypoxic and sometimes they're not, dependent on the oxygen consumption and gradients in culture. But we eventually adjusted all that and found, somewhat to our surprise, that all cells do this. And that was an important moment in my life, but we eventually realized that this would be rather significant. I have an interesting anecdote for those who have difficulty publishing their work. I felt that the work was very important eventually, and I would therefore, since there was no electronic submission, I drove in my old car to the Nature offices in Little Britain. It was Maundy Thursday. I was particularly anxious to get to the editor, explain the importance of the work before the Easter break, on the basis that I felt sure that they would publish it immediately. It was very interesting. They're obviously used to people doing this. I arrived at 10:30 in the morning and explained I need to see an editor, and the secretary says, well, they're all at lunch. You know, it seemed rather improbable at 10:30 in the morning, but there we were, and we didn't get it to them, nor did we persuade them to publish it. Eventually, the thing in Oxford, if something like this disappointment happened, was to go and see David Weatherall and sit in his office until he did something helpful to support your cause, which he did in making this submission, which was not the journal of first choice. But that did establish that this was not a private system to EPO, but it was general, and it must have other uses, we believe. As you've heard, Greg Semenza identified HIF, the transcription factor that's responsible for that, and we identified the first non-EPO HIF genes as a rather surprisingly phosphoglycerate kinase. It was very difficult to persuade people at the time that these two quite different physiological characteristics, this one regulated 3 orders of magnitude by severe anemia, and this one barely regulated at all, were in fact joined to the same system. But that turns out to be true, and we now have successive waves of understanding of the HIF system, moving from things that have a direct effect on oxygen balance to perhaps rationalized as repair of hypoxic damage or co-option of hypoxia signaling into quite different types of biology. It's not really clear whether this is a new physiology of hypoxia or whether this is co-option of the hypoxic regulatory system for other purposes. And I'll go into Darwinian biology co-opting things and the difficulties that makes for us trying to understand biology and make drugs later on. But we now understand that this is extraordinarily broad system. Practically every aspect of cell biology will be regulated by this system, as you might anticipate, given the importance of oxygen to biology. But the task at the time was to move upstream from HIF to what we believe will be the next part in the signal transduction mechanism, and we took the HIF sequences and spliced them onto artificial transcription factors to see which bits would confer this property of regulation by hypoxia and, interestingly, cobalt, also ion chelation. And you can see that this piece does, but these don't. That work established really that there were two types of regulation through protein stability and a type of regulation that was similar but not dependent on protein stability. And of course we knew that something in these sequences must interact with the signal that was generated by this oxygen sensor. So everyone knew that protein phosphorylation was the means of signal transduction, so everyone was looking for the phosphorylation sites. And we mutated every phosphoacceptor in this region. It made no difference at all. That work, as you can imagine, was also difficult to publish. The reviewers, I got their comments, well, this is entirely negative work. We wonder how the authors had sufficient laboratory resources to do all this negative work. So, of course, that wasn't very impressive for the editor. But actually, it was quite important because it excluded the main hypothesis as to what the signaling mechanism was. It was something other than protein phosphorylation. The next clue came from clinical medicine that kidney cancers, which are defective for this VHL tumor suppressor, often highly angiogenic. They sometimes produce EPO, and in the familial syndrome are associated with overgrowth of blood vessels elsewhere. And that led a number of investigators, including Bill Kalin and Rick Klaus and others, to propose a link to hypoxic signaling. And to note that some of the genes that we and Semenza and others had described as hypoxic sensitive were stabilized in VHL defective cells, but they didn't make the connection to HIF. We were able to do that, and actually it was rather straightforward. The HIF in the VHL defective cells is stabilized, whereas when you put HIF back in, there is this enormous regulation by oxygen. So why were we able to do this? And this is a lesson for the young people in the audience. If you want to lead a field, you must take the trouble to make your own antibodies. So we were the only group that had HIF2 antibodies, and it turned out the cells that were being investigated only had HIF2. So people who had just the HIF1 antibodies drew, as did we, a completely blank and puzzling blot. But this unlocked the problem, connected VHL with HIF, and that's the system that oxygen promotes association of HIF with VHL. And VHL is a ubiquitin ligase which destroys HIF in the presence of oxygen. And when it's not there, the HIF builds up and does that transcriptional response. So the question was what was the nature of this association? Of course, that enabled a biochemical approach. As you've heard, I'm a nephrologist, not a biochemist. And the other lesson is to try to pick a field which is not absolutely cluttered with people. Since we knew little biochemistry, we muddled our way through this without too much sophistication. It took quite a long time, could have been done much more quickly. But eventually, we learned that the extract was heat labile, it needed oxygen, it didn't need ATP, consistent with it not being protein phosphorylation, it did need iron supplementation. We were able to implicate by mutation of the HIF peptide, this is a capture of VHL by HIF peptides, that this proline was important. And we were able to do mass spectrometry on the peptide that revealed an extra 16 Dalton shift on this proline-containing peptide. So those of you who know a bit about mass spectrometry will know that this sort of thing is extraordinarily unreliable. There's a lot of oxidation. So the killing experiment was actually to make synthetic peptide. It cost a lot of money, took ages to come, but that's the result, which I refer to as a Sunday morning experiment. You'll see that when the hydroxyproline was there, we no longer needed treatment with that extract. Sunday morning, because the postdoc concerned rang me with the result on Sunday morning. That means, unusually for my lab, the experiment was conducted on a Saturday. It was considered sufficiently important to read the result on a Sunday morning, which is doubly unusual. But what is triply unusual is that the result was positive. Of course, these hypotheses usually go down, and the postdocs come in on Monday and tell you the miserable news that your hypothesis was not substantiated. But that one was, and it was important since it implied a prolyl hydroxylase, which are oxygen-splitting enzymes. They require iron, and they're inhibited by cobalt, in fact. So that was the system so far, prolyl hydroxylase, producing that unprecedented signal modification that promotes association with the VHL ubiquitin ligase. The question is, what is the enzyme? And this just illustrates the way science works. This is my friend, colleague Christopher Schofield, who had been working on antibiotic synthesis, and he obtained these crystal structures of the 2-oxyglutarate-dependent dioxygenase family, which also includes the collagen hydroxylases, and was able to make predictions across the human genome as to candidate prolyl hydroxylases. And sure enough, one of Chris's predictions came good. Again, antibodies are essential. We had identified the C. elegans HIF and made antibodies to it, so we could readily receive these mutant worms from the well-organized C. elegans community. And we see that the wild-type worm regulates its HIF, whereas these worms that were mutant for a hydroxylase or a proposed hydroxylase that Chris had predicted were totally lost in relation to hypoxic control. So this suggested that indeed EGL9 was the oxygen-sensitive hydroxylase, and that turned out to be the case. In mammalian cells, there are 3 of these enzymes, 2 sites of hydroxylation, and then HIF is also that non-proteolytic regulation. Murray Whitelaw's group established a different type of oxidation, asparaginal hydroxylase, and with Chris, we and others identified that enzyme as a protein that Greg Semenza had first identified as factor inhibiting HIF, FIH. So there are 3 prolyl hydroxylases and 1 asparaginal hydroxylase in human and most mammalian cells, and there's oxygen-splitting enzymes which perform these reactions, and in the presence of oxygen leads to the degradation of HIF and the blocking of its activity. So, as Joachim said, that's an unusual and, in fact, unprecedented signalling modification. So we've been interested in the relation to earlier biology. And, in fact, all 4 eukaryotic kingdoms, interestingly enough, use this odd system of protein hydroxidation, protein oxidation and proteolysis to signal oxygen levels. But we're not as yet clear about the interrelations here. The 2 closest, the protists and animals, use very similar enzymes, but they have a different system, whereas you've heard in animals we hydroxylate HIF, the transcription factor. In protists, they hydroxylate the ligase, and that leads to the regulation of culmination factors that control the association of single cells into multicellular aggregates. Plants and fungi, yeast apparently use different systems. But they both relate to protein oxidation and degradation. Plants controlling ethylene response factors by this system, and at least in fish and yeast, the sterol response. Now, we've been interested in what the relationships might be to the HIF system, and one of the lines we've pursued is to look at the enzymes most closely related to the prolyl and disparaginglyl hydroxylases in the human proteome. Interestingly, both the prolyl and the disparaginglyl hydroxylases are closely related to other enzymes, both of which are involved in ribosomal hydroxylation. This one, OGFOD, is in fact in the ortholog of the enzyme in fish and yeast, which regulates the sterol response. And it appears there that the production of the ribosomal subunits in some way interacts with the synthetic cellular material that is regulated by the sterol response, somehow coordinating ribosomal satiety with other synthetic mechanisms. If we look back to the prokaryotic enzymes, the closest relation of the prolyl and disparaginglyl hydroxylase are both, again, concerned with translation. So although we don't understand it, there seems to be some evolutionary link between oxygen satiety and protein synthesis, which of course is a very energy-consuming reaction. I'm not going to go further into that at the moment, but want to return to the HIF system in animal cells and talk about the relation to integrative physiology. So this system is present in its assembled form only in animal cells, but it's present in all animal cells as far as we can see. So all animal species have a HIF and a PhD. Some of them have two HIF prolyl hydroxylase sites. Some of them have the asparaginglyl. And then at the base of vertebrate evolution, you get these gene duplication events, which give you more PhD, more prolyl hydroxylases and more HIFs. Now, of course, in higher animals, these are the ones that use the heart, lungs, bloodstream for specialist oxygen delivery systems. So the question is there any physiological specialization? Does the increase in the number of these enzymes and systems have a relation to the development of these physiologically interesting and medically interesting systems? And that's what I just want to go through that interface in the next section of the talk. How do we go from this, possibly the ultimate in reductionist biology, a simple oxygenation of the carbon-4 on the prolyl residue to create an alcohol group, which then hydrogen bonds to the floor of the VHL pocket. That's a switch that apparently is able to regulate all this oxygen homeostasis, which we need to control in higher organisms. So I'll consider three aspects: integrating biochemical signals, integrating developmental signals, and integrating physiological signals as exemplars. The biochemistry first. It's interesting that a lot has been written about the potential for these enzymes, which also coordinate a Krebs cycle intermediate as co-substrate, and have a labile interface with iron, which is potentially redox sensitive, for these additional inputs to modulate the oxygen sensitivity. And indeed, some people have proposed that it is the production of oxygen radicals, not dioxygen per se, which is the sensed entity. We feel otherwise, and I'll show you some of our data. But it does raise interesting biochemical questions. So we've got three sites of hydroxylation. Question is, what is their relative sensitivity to hypoxia, and what is their relative sensitivity to oxidant stress, if that is the regulated principle? This is a summary of our data that in relation to hypoxia, there is an order that the N-terminal prolyl site, then the C-terminal prolyl site, then the asparagine have that ordered sensitivity to hypoxia, which is the exact reverse of the sensitivity to hydrogen peroxide. So we do not believe that these stimuli are equivalent, and believe, therefore, that there is a potential for them to interact. There's one piece of data that might be true. If we look at the interaction here between hypoxia and peroxide, which is not necessarily a physiological stimulant, we can see that in peroxide-treated cells, we can augment the hypoxic response. We believe that is due to the sensitivity of this asparagine hydroxylation, and the evidence here is that when that enzyme is knocked down, we do not see that augmentation. Now, that's just one example, and it's obviously a set of conditions in the tissue culture which we arranged to see that. So it sort of exemplifies a potential rather than tell you that this is actually the way you work physiologically. But this is the proposal, which I don't think we can confirm or refute at the moment, that there has to be a matching of the rate of hydroxylation with the rate of degradation. And when those systems are on range, hydroxylation will be rate limiting, and therefore the system is oxygen sensing. If we're at the top of the range, of course that wouldn't be the case, nor would it be the case at the bottom of the range. But what we might propose, just as a possibility, is that under conditions of high oxidant stress, some of the relevant enzyme is inactivated, and that changes the in cellular kinetics to be differently oxygen sensitive. I just put that as a hypothesis, since we lack an explanation at the moment as to why cells working at entirely different oxygen tensions can deploy this system for their oxygen sensing. I'll leave that hanging there. Development. So when we found that this system was general, we immediately believed it would be true in yeast and all those other primitive animals that I've said don't have it in this integrated form of the HIF-PhD system. We also believed that it might control cell size, would control things like implantation, primitive developmental processes. And that again turned out to be not true. And this is an experiment that we did with Pablo Wapner in the fly. This is the fly tracheal system. You will know that the fly gets its air through oxygen through a system of air passages akin to the mammalian trachea. The fly doesn't carry oxygen by the blood or have the heart system, but it has this primitive lung-like setup. And that's the wild type. If we make a PhD, a hydroxylase mutant, which will mimic hypoxia, we see that, as is the case with hypoxic flies, that system becomes greatly complicated. You make more air passages, so that all makes sense. If you make a double mutant of the PhD and the HIF, there's just one of each in the fly, comes back to normal. So at least in the primitive organisms, the developmental plan is already sufficient in a way for oxygen homeostasis, and this system works on top. That's not necessarily true in higher animals, but in higher animals it seems that early development is compatible with oxygen homeostasis, but in later development HIF is induced and it has a role in development. This is not our work, has come from one of the Zurich groups who made a knockout in the heart, which develops zones of hypoxia, induces HIF, and it would appear that that's important in later stage cardiac development, because in the HIF knockout, they fail to develop the ventricle in in the normal way. So this system is built on top of an already compatible body plan, which is enough to enable a fly to develop normally in the presence of oxygen. It's not enough for later mammalian development. So an interesting interface with development. I said I'd talk about physiological signals, and the one thing that we haven't explained here is the rapid regulation of certain physiological things like respiration and pulmonary vasoactive responses, which happen in seconds, clearly not due to this system, which is dependent on transcription. But how do they interface? I want to go back to Haldane, to that thing, because there's something remarkable. It's worth reading the paper. It's on 55 pages in J Physiology, 1912 or something. And he wanted to see whether the carbon dioxide level went back. But what actually happened, you see, is we carried on going down. Now, it's a 56-page paper, so there are 2 pages devoted to this. They don't sort of say, well, this will cause trouble with the referees, so we'll discount the data for the moment. Well, this happened in all of the subjects, and we're completely puzzled by this. It was quite reproducible and entirely unexpected that actually respiration, rather than correcting, increased with a few days altitude. And that is the phenomenon which you will know of acute acclimatization, that pre-exposure to hypoxia increases the sensitivity to acute hypoxia when applied in minutes. So the question is this HIF, and how does it relate to the system I've described? And we set up a mouse system to study this with plethysmography, pre-exposure for a matter of 7 days to an altitude quite severe, about 5,000 meters, has a very large effect on the respiratory sensitivity. We use standard knockout technology to inquire whether that was related to HIF1, which it apparently is not. But in the HIF2 knockout animals, we saw a dramatic loss, almost back to baseline, of that enhanced sensitivity. Those effects were recapitulated in another response that the carotid body, which we believe is underlying that ventilatory sensitivity, makes to hypoxia. It ordinarily produces this large proliferative response here measured by BRDU incorporation. But in the HIF2 knockout animals, that is almost completely ablated. So here is an example where what we believe to be a modern form of HIF2, is involved in a modern aspect of oxygen homeostasis. We're a little bit surprised by the contrast between HIF1 and HIF2 there, until we looked at the expression patterns here by RNA scope. And HIF2 is about 50-fold more expressed in the carotid body. Here, you see it's much more strongly expressed than any of the surrounding tissues. So there's something extraordinary about the expression level of HIF2 in the carotid body, which is way, way more, as far as we can tell, than any other organ, and is important in the tuning of respiration. We went on to knock HIF2 out in the type 1 cells. That was the ones that were showing the HIF2, and sure enough, there is the reduction in the response after acclimatization. And looking at the type 1 cells, there's some rather remarkable differences. So people who've worked on the carotid body will know this, that after exposure to hypoxia, the vesicles change, and there are 4 different powers of electron microscopy here. You see the high power demonstrates this eccentric vesicle So what happens in hypoxia? The dense core vesicles reduce in number, but they change in form in this rather dramatic way. And you'll see that that does not happen as far as we can see in the HIF2 knockout. So HIF2 is doing something in the type 1 cell, which is the oxygen-sensitive cell, we believe, that impacts on the rapid response. That's the secretion of these vesicles in response to hypoxia. So specific physiological functions owe their operation to specific HIF isoforms. That seems to be true. But a really important issue in the field now is, can we make a drug? Can we use this system, since hypoxia is so prevalent in human disease, to deal with one or other disease where we would generally like to intervene on a specific physiological function? So in other words, can we go backwards? What would be the specificity of a particular HIF isoform? We know that the specific physiological function, control of breathing, for instance, it would also be true of erythropoiesis, has a specific relation to HIF2. So we've done a different type of experiment here. It's not a physiological thing, it's a transcriptional experiment. What are the targets of these different HIF isoforms, HIF1 and HIF2, across the genome? And you see here that how we do it. This is a chromatin immunoprecipitation. And this locus is both a HIF1 and a HIF2 binding site. And we see that they sometimes overlap like this, but sometimes they're distinct. If we look, interestingly, at the totality of those across the genome, they have an identical consensus sequence that they recognize, identical, but they're quite differently distributed in that the HIF1 is rather proximal, rather close to promoters, whereas the HIF2 is rather far away. And that, again, is a modern characteristic. The difference between human beings and yeast and things, not mainly the number of genes, it's the number of complexity of the control sequence. So we have a lot of non-coding DNA, which is principally concerned with regulation and is a modern, relatively modern characteristic. So HIF2 looks like a biochemically modern isoform. What would happen then is, since these bind the same sequence, if we knocked one out, and this is the plot of the chip-seq signal in wild-type cells versus a knockout cell. And you see that the knockout of neither isoform changes the distribution of the other. They're like two pianists playing on the same piano, using in part the same keys in a wholly independent way. Now, that's a bit difficult perhaps to understand, but there'd be kinetic reason. The implication is they're only in contact with the DNA for a very short period of time, so they don't interfere. So there are quite distinct properties of these systems. Could they then be drugged in a distinct way? That would be possible if it were done. Would they have a defined physiological function? And the answer is unfortunately no. That the system appears to be used over and over again. So not only HIF2 supports respiration, erythropoiesis, a whole raft of other things. And what we would like to do in drug development, isolate a biochemical property which would have a single physiological reflection, is not really possible. They each have HIF1 and HIF2 distinct targets, but they're all intermingled, so we could neither pick off HIF1 nor HIF2 and have a clearly distinct physiological effect. They have, as you might imagine, different associations with other transcription factors, which we believe are probably the associations controlling their selection of particular hypoxia response elements across the genome. But the fundamental issue is this, that Darwinian biology, which has used the same system over and over and over again, for physiological functions is fundamentally at odds with rational drug design. It just is very difficult to take a thing apart and get what you want. Given all this, given the enormity of the HIF system, how could it be that what turns out to be the case is it possible to get a relatively isolated erythropoietic response to an inhibitor? But yet, I think if you'd asked all the scientists in the field, would this be possible in 2001-2, we would have all said, no, you're joking. This is a pleiotropic system. You will not be able to isolate and treat anemia by activating HIF. Yet, 15 years down the line, that's exactly what the companies appear to be showing, that these are essentially all showing similar types of experiment, replacement of EPO or treatment of renal patients with a prolyl hydroxylase inhibitor, increasing the hemoglobin level without obviously doing anything else. So this is the moral and the finish of the story, really. Reductionist biology can take you so far. So you can't always get what you want. You can't, in other words, find a reductionist principle that isolates the medical effect you want. But if you try, sometimes you find get what you need. The prescient words of Sir Michael Jagger, of course, he knew a little bit about drug discovery. The principle and moral of the story is, though, Darwinian biology is so confusing that there is an element of empiricism required in developing your drugs, and that's exactly what you see here. The concern is that the massive phase 3 trials that are now in progress, if they do not deliver ultimately the result, which is a drug superior to EPO, they will be abandoned without all the trial and error and testing that would be necessary to tease out empirically whether you can get an important medicinal effect from this system. Now, I could write you a really nice essay as to why these drugs apparently can work on EPO and not other systems. I can tell you that the drugs are often concentrated in the liver and kidney. I can produce really interesting arguments about the countercurrent mechanism of exchange of oxygen and how that might interface with drug distribution and the effect you saw. I can show you that the EPO effect is 3 orders of magnitude, so for whatever reason intrinsically rather sensitive. And I can explain to you that the red cell life is about 120 days, so essentially it can act as an integrator of the system and therefore enable through careful dosing to give you an effect on the red cell, not on other systems. But let's be sure, that is all post hoc. I did not and could not, nor could anyone else have predicted that if this result turns out to be true, it would be possible to isolate the erythropoietic effect from other physiological systems. So that's the summary slide. Haldane's expedition was famous, of course, for the proposal of the secretion of oxygen, which was untrue. So in that expedition, everything was done experimentally. All the measurements were beautifully correct, but all the deduction as to mechanism was wrong. We need to define molecular mechanism at the molecular level, and that's what the story of EPO and oxygen sensing shows us, since practically everything had been proposed on the basis of conjecture except the correct type of mechanism. Having learnt that lesson, we shouldn't be arrogant. We need to define integrative physiology at the physiological level and define clinical effects of drugs at the clinical level. Nothing much else, sadly, will do, and that is my final word for the support of physiology and the support of empiricism in the clinical development of drugs. This is my group, to which I'm immensely grateful, on a sunny day in Oxford, and we've enjoyed great support over the years from the Wellcome Trust, recently from the Ludwig Cancer Research Institute. Thank you very much.